Okay, I'm going to go ahead and get started. So, go ahead, Dan. I have a couple announcements to make. All right, so a couple announcements. We identified a couple adding mistakes on the exam. So again, just make sure that definitely look through your exams and just double check to make sure it's added right. There's a couple examples for of people, like a common mistake, like you can think that people would make, right? If there's like a multiple question answer, there's a couple instances where clearly it was like the bottom score that ended up on the first, so they didn't tally that whole page. And that was like, I think we saw like one or two examples of that. So double check, make sure all the points that are there are the ones. I know you guys are generally really good at that, but I'm just going to emphasize that again. Just look through your exam. There's a lot of you guys here. Mistakes can happen. So just make sure specifically that there's no sort of adding mistakes. I also found out, just as a heads up, the last quiz, I think Friday's section, I'm not sure what happened. I don't know if there was miscommunication, but one of the questions was graded completely wrong. And so it was this question of whether the molecule was aromatic or not aromatic, and I'm guessing whoever was grading it just had a brain fart or something and was grading it as aromatic. So some of you will probably get points, others will end up losing points. So that's just a heads up if suddenly you notice your quiz grade changes. And again, it's the Friday section. So, all right. So moving forward, what we're going to talk about throughout the rest of the class here, quiet it down. We're going to talk about a little bit of chemistry. There's really not too much left for carbohydrates, but I think that there's something really interesting and fun that we can talk about that I think has human health relevance. So I thought it would be of interest to you guys, and it also represents something that I personally have some experience with. So at least I can give you a perspective of research that's being done in organic chemistry groups associated with sugar chemistry. So with that, oh, sorry, one more announcement. Friday, again, is going to be the first of the next series of quizzes again. So if you've got recitation this Friday, expect a quiz. What will be on it, again, because we haven't had too many classes since the last exam, if you're up this Friday, basically carbohydrates are fair game. So the stuff on Wednesday you don't have to worry about, but everybody else does. So because everybody else will have the additional weekend. So if you're Friday, today's class and last class. So mainly what you're talking about is sugars, being comfortable drawing and interconverting between the Fisher projections and the sort of 5 or 6 membered ring structures. Some of the chemistry that we've learned on sugars, so just recognize some of the chemistry that can take place on them. We'll talk about a few extra reactions today based upon them. And we'll talk a little bit about stereochemistry naming. I forgot to go over that, but there's just something specific about stereochemistry naming for sugars that I think is worth mentioning because it's fairly common. And so those are the sort of things that will be responsible for sort of people on Friday. People that are following week will also be responsible for the stuff that's on Wednesday. Again, I always like if people have an extra weekend, that's a significant amount more time to study.
Will metals be covered?
No. No, but there'll be heavy emphasis on it on the final. So, yeah, no, just everything since the exam. I just like to keep moving forward, and since that was somewhat covered on the exam, it won't be on the next quiz because I want you guys to sort of catch up on it. Okay, so sort of in just terms of reactions of sugars, and I'll actually later on today I'll discuss like actual chemistry of sugars because it's kind of interesting, and I think it's worth putting in perspective. So I'm going to go off book in a second, but just sort of the chemistry that your book discusses. So again, one of the things that we sort of have discussed is these sort of hemiacetal, cyclic hemiacetals, right, can exist as an open closed form of these molecules. And so what you end up finding is a lot of chemistry. I think the whole point was that you end up finding that a lot of chemistry that you can do sort of on sugars can also be represented by chemistry that's in general on aldehydes. And specifically, as we sort of discussed last week, anytime you have a molecule that is, say, a sugar, that has an aldehyde in it, so again, these are the sort of aldoses, right? What we know is that you can take these molecules and treat them to sort of the Tollens test. So again, this is the sort of silver 2 test. This is Tollens. Again, this is the thing that we showed the sort of video of. So it's basically that cationic gold being converted to sort of gold 0, which then becomes very obvious. And it's, again, one of those things that was sort of done classically to sort of be able to determine structures. And again, the sort of net result in this process is that you get a carboxylic acid. Yes?
Why do we work better with tertiary carbon? Like, in the lab, I talked about how tertiary carbon react very quickly with secondary ones and primary ones, and what react with common stuff.
It's an aldehyde. It's a reaction of an aldehyde. I'm not sure what you're talking about.
With alcohol, sorry. Does it also react with alcohol?
So I'm not that familiar with exactly what you're talking about. So when you're, so is this for like the unknown lab? Yeah, I'm actually not that familiar with that. So what are you saying? But maybe I can give you an answer.
It does react very quickly with tertiary and then
Well, it shouldn't be able to react with tertiary. Yeah, because you'd have to do an oxidation, right? So maybe it's a different process. Yeah, show it to me. Maybe we can talk about it afterwards. But yeah, I mean, if you're a tertiary alcohol, it's kind of difficult to oxidize up, I would think. So anyway, if the carbonyl is somewhere else in here, but again, I could be wrong, so show it to me afterwards. Now, if you're doing this with a sort of a molecule that's already oxidized, like a ketone, right, you're not necessarily going to be able to, right, so if it's a ketose, in general, right, if it's a sort of a ketose like this, you're going to get a sort of a negative tolerance. Okay, so in general, for ketoses. The only time when you'll occasionally see Tollens, I guess, according to the book, time where you can sometimes see Tollens tests give sort of positive results on ketoses is when you have the sort of ketose adjacent to the alcohol at this position. And again, we can draw a mechanism that sort of explains why. So if you have a system like this, you in fact can undergo Tollens test, I guess. And the reason behind this is that what you can actually do is you can effectively do a tautomerization followed by another tautomerization. Where if we just sort of isolate this part up here so that we can see what's going on. You can basically go to the tautomer. So color. So what we're talking about here is, right, is that alcohol there. Basically, this is just a tautomeric form here, right? And then now when you're at this stage, you can go to the tautomer the other way and actually get to the aldehyde. So, again, this is kind of interesting. So I guess just under the conditions for the Tollens test, it allows for this. And so You can also get active tolerance tests for this sort of polymerization process. So again, I'm not that familiar with this process, but at least mechanistically it makes sense, and your book describes this. So my understanding is probably that just under the conditions for the tolerance test that allows for this sort of polymerization process that will effectively allow you to go from here to here. And again, this is going to be not a resonance, but a reversible process. And again, once you're at that aldehyde, then that aldehyde can in essence oxidize up. Okay, so this is sort of the important point about the tolerance test that you guys should sort of, I guess, know. Okay, and again, I think the major value of the tolerance test was when people were trying to differentiate between different classes of carbohydrates back in the day to know if a molecule was a ketose or an aldose. So again, it can, if you get a negative tolerance test, and what that means is that you have a ketose, but more than a ketose, the ketone can't be sort of here. So it's almost better to think about it in the opposite direction. It's like the tolerance test negative basically means you have a ketose somewhere buried in that sort of side chain, okay? Such that you can't do that. Right, or you may just not have sugar, right? Okay, so the one other reaction that the book discusses in terms of chemistry of sugars that can sort of be valuable is the sort of use of periodate cleavage, right? So as you guys remember previously, if you have molecules that have diols, right, this is from last semester, or sort of first semester organic chemistry, you treat it with HiO4, water, right? Sometimes you'll see it as H3IO5 or something like that. But anyways, periodic acid and sort of water. If you remember this process, you go through sort of an oxidized sort of form here, where you effectively break this bond by basically putting electrons back into the iodide, so it's an overall oxidative process that leads to two sort of aldehydes. Okay? Now, again, we've sort of already learned this, so where was this used in sort of figuring out the structures of sugars? It was particularly valuable in terms of determining whether or not molecules were sort of 6-membered rings or 5-membered rings. So if we had a, say for example, horrible rings. So in general, if we took a sugar, right, and then we sort of treat it with acid and yeah. What's that? So it's metal based. So basically what's happening is you have a very high oxidation state iodide. Once you generate this complex, it breaks down to take the iodide from a higher oxidation state to a lower oxidation state. So you can think about this as being a high oxidation state. Currently it's 1, 2, 3, 4, 5, 6, 7. It's iodide 7. That's really high in oxidation state. Once it breaks down, it goes to iodide 5. So I may have, pretty sure this is roughly right if I drew it right. But the bottom line is the same. Basically, you're going from a high oxidation state to a low oxidation state. Throughout that process, what's happening is you're losing a couple electrons in that diol, right? So oxidation is loss of electrons. You're saying, how do you go from here to here? Yeah, but you got oxygen here too. Well, unless you lose water. Okay, so, all right, so here's the general sugar, right? And what can happen is if you've got, again, stereochemistry doesn't matter, again, based upon the mechanism. You might think that you might need syn, that might make it faster, but you can sort of exchange out and still make it. So it's not really important to think about the stereochemistry, although it's going to be faster if there's syn. But the important point here is that what you have is a couple of, in a 5-membered ring, you have a couple of diols, Yeah, you're generally not responsible for metal-based mechanisms anyway, so. Except for those that I told you guys you're responsible for. So don't worry about the mechanism of this. So what you see here is there's effectively two dialdehydes, right? Or sorry, two diols, right? And so if we sort of go through the mechanism of this, what's going to happen is you're going to break that oxygen, those sort of carbon-carbon bonds, and you're also going to break this carbon-carbon bond. Okay, so you can imagine that if in this process, what happens is when you do this overall oxidation process, what you're going to lose is effectively, I think it leaves this formic acid. This group will leave this formic acid, or some oxidized carbon source. Could be like CO2 as well, but I think it's formic acid that leaves it. But the more important point is that you effectively break both these bonds, and what you're left with is sort of smaller molecule here, right? So of the two areas where you can imagine having 1,2 diols, you sort of get this cleavage. Again, if you go through the mechanism, you can, if there's still the other alcohol there, you can go through the sort of hydrate. But this is a general process that's going to take place. So if you have a 6-membered ring, what's going to happen is you're going to start losing some of your carbon sources. So if you do elemental analysis or something, again, this is sort of an old-fashioned way to sort of do structure determination by chemical modification. Right, or perhaps you can get mass spec or something. You sort of make that modification, and now you've got that. And what you notice is that if it's a 5-membered ring, the same sort of thing happens with a, say, a 5-membered sugar. There. In terms of 1,2 diols, in this case, you're only going to have a single one, right? So even if we have, say, an alcohol or something over there, the only 1,2 diol that you have in this case is this. So if you do the same thing with this compound, if you do the periodate cleavage of this, you're not going to lose the sort of formic acid. You're just going to sort of open this up, and so your mass isn't going to change that much. Okay, so in this case, you're just breaking it once, you don't lose that other thing. So apparently back in the day, this was used as a way to sort of determine whether or not a cyclic structure was a sugar of a 5-membered ring or a 6-membered ring. And that's, again, I think a lot of the chemistry within sugar chemistry, within this chapter, discusses that. Now, what I'll get to in a little bit later on, just to sort of show you guys the state of the art of sugar chemistry, is a little bit more sugar chemistry within synthetic organic chemistry, just so that you guys can get a feel for how difficult making sugars are in this day and age. But again, the chemistry that you primarily learn through this chapter really has its roots back in structural determinations of sugars. So not too dissimilar from what you guys are doing in your lab course in that way. Okay? All right, so what we'll do quickly, I just want to talk about stereochemistry of sugars for a second in terms of naming it. I'm not a big guy. Yeah? What's that? Oh, the formic acid? Yeah, you effectively, I'm not sure how they exactly monitored it afterwards, but you would see a significant, in this case, let's say you did mass spec, for example, right? You took a mass spec of the starting sugar. If suddenly the mass of the ending sugar was simply minus 2 protons, right, then you know that it might be an indicator that it's a 5-membered ring. If you did mass spec in this case, right, you take the mass spec of the starting material, then you take the mass spec of the product, and now you see there's a big drop-off in terms of not only do you lose the 2 hydrogens, but in addition you lose an equivalent of carbon dioxide as well. So basically CO2H. So how specifically back in the day they actually determined that, it might have been elemental analysis, but at least you can rationalize it just thinking about mass spec, for example, which should make sense. So it's more about structural determination. Okay, so I do just want to talk very briefly about naming, just because you guys will see these sort of letters of D and L in the sort of literature, and so I think it's worth it, since you'll see sugars called D or you'll see sugars called L, it's probably worth it to sort of understand why they're named this way. And in truth, the name is, like a lot of things, sort of has its origins really in terms of, actually a lot of this has to do with sort of characterizing molecules and understanding their structure. They just needed to give sort of names to different things in order to classify them into different groups. And so through these sort of breakdown processes of sugars, a lot of times what would happen is that sort of end unit that you can have, right, is get this right. It's sort of glyceraldehyde. Okay, so this is sort of the glyceraldehyde, right? This, as I've drawn here, is D-glyceraldehyde. And if we draw the other enantiomer of it, it's L-glyceraldehyde. And the question comes down to sort of how do we name these? Now, it turns out that it was simply based upon optical rotation. So if you sort of shine plain polarized light at a molecule and it's one enantiomer, usually it'll polarize in one direction. If it's the other enantiomer, it'll go in the other direction. So chiromolecules will effectively bend plain polarized light in one direction or another based upon the chirality. And so a lot of the names, this sort of D and L, really came due to the sort of way that they bent the light. So that's not really what's important. I think what's more important is perhaps for you guys to recognize what would be a D sugar and what would be an L sugar in terms of the structure of them. And so this is really where it all starts from, is glyceraldehyde. As we'll see when we build up sugars, the naming system really sticks with this. So you sort of build up, and it's really the glyceraldehyde where this sort of starts from. So I thought it'd be worth it to sort of revisit these guys in terms of stereochemistry and try to refresh on, is it R or S? Because it might have been a while since we sort of thought about R or S. And so let's ask the question, is D-glyceraldehyde R or S? And then likewise, is L-glyceraldehyde R or S? So as I'm drawing it, why don't you guys think about it a little bit? So for D, I have a little, especially when it comes to nomenclature, sometimes you just need some type of way to remember them, and so I usually come up with silly ways to sort of keep these together. But again, if we want to sort of draw this, again, what you have to do is you have to put the proton in the back, so what we're going to do is flip this upwards, put the proton in the back, right, and when we do that, what's going to happen is you're going to have the aldehyde over here. Let's see, did I do that right? I believe that I did. Anybody want to correct me? Okay, so, what's that?
You can just draw it the way you normally do it, just put the hydrogen on the back, and that's going to be it.
Yeah, no, I mean, this is it. I like drawing it this way. So, okay, so this would be 1, this would be 2, and this would be 3, right? I always like putting the proton in the back. That's why it helps me get comfortable redrawing molecules and so forth. Okay, and so if this is our molecule, it's R, right? So 1, 2, 3, right? Again, the 2 takes priority, so this is R. Now here's the awesome trick that I came up with when I was a graduate student, or actually when I was undergrad. There's D, now it's R, right? So I literally take a D, and you can convert it to an R. And I don't know why, but that's how I always kept the 2 in line. And then if you sort of forget, the other one would be L, and yeah, you can sort of turn that into an S. So that was the sort of way that I at least helped remember what D was and what sort of L was in terms of R or S. Okay? Now, again, I'm not going to go through it per se, but what you can sort of recognize is if you start from the bottom part of the sugar and you build up, there's going to be all these different names of sugars. but the sort of DL classification is based upon the sort of glycerol portion of the molecule. So the sort of bottom aspect to it. And again, you guys can read through the book, but I just thought it was worth it to sort of bring up the DR LS classification. All right, I want to get into more interesting stuff. And hopefully it hasn't been abundantly clear how bored I am of this specific material. But I think sugars can actually be really cool, and there's some really interesting and fundamentally important aspects of sugars that aren't in your book. Well, they touch upon some of these things in the book, but I thought it would be more fun to sort of emphasize them a little bit more, and so forth. And so the two things that I'm going to focus on throughout the rest of the class today are really going to be sort of antigens. Anybody know what an antigen is? Just out of curiosity, how many people know what an antigen is? Because I didn't know what an antigen was until I was a postdoc. All right, so maybe about a quarter of the class. So an antigen is basically a molecule that an antibody binds to. Okay, so antigens are things that are, they're basically the things that antibodies recognize, bind to, and effectively help serve as that homing beacon for the rest of your immune system to sort of go to town on whatever the pathogen is that your body wants to get rid of, or neutralize it, or anything else. And sugars actually play a really, really big role in terms of sort of the human immune response. Because as it turns out, different types of organisms, in terms of cells, in terms of sort of protein expression and so forth, different organisms will often have very different sugar-based antigens displayed on the cells. And that's one of the ways that sort of your immune system uses to sort of recognize if something's foreign or not foreign is sort of that telltale point. So does that make sense, right? It's kind of really an interesting thing about nature if you think about it. So again, if you have certain sugars displayed on your cells, right, then your body is going to recognize anything that has those same sugars as being okay, it's safe. But the moment that your body sees maybe a different sugar that your body's not used to, it will bind to them, it will recognize them, and it will try to clear them to get rid of them. And there's a couple places where this is sort of really important, and I think one of the critical places in this sort of, and your book does discuss this a little bit, but I didn't think it was that great, is the sort of ABO blood system, right, which it really has all of its bearings in terms of us, right? So different species have different types of sugars, but even within sort of humans in general, we will have different sugars within our species, and that's really important, right? Because that's the whole reason why when you go to give blood, they need your blood type, because they need to know what type of sugars you're displaying effectively, so that when somebody else gets that blood, that they are also recognize your blood as not being foreign, right? So there's a couple aspects within sugar-based antigens that are important. Matthew? So yeah, I'll go through it. Yeah. So all right. So if you guys sort of know groups, right? There's really A, group B, group AB, and group O, right? And really what we're talking about here is your red blood cells, right? It's the red blood cells. Okay? And so what you have, right, is you have your red blood cells. Actually found the figure that I'm about to put in here, I kind of found on Wikipedia. I'll tell you, Wikipedia, I remember when I was like, everybody says don't use Wikipedia as like a resource for stuff. It's true to an extent, but it's actually fantastic if you don't know anything about something. Wikipedia can be great just as a sort of a first over, but then you certainly want to go back and make sure that everything's there is pretty accurate. But it's at least if you don't really know about stuff, if you're reading something fairly preliminary, Wikipedia can actually be great, but don't go citing it in papers because it's unprofessional. All right, so if you imagine sort of antigens, right? Okay, so for antigens, let me try to get this right for you guys. Okay? Let's just discuss the different sort of blood cells. So, again, as we sort of discussed, this really all has to do with sort of the sugars that are being displayed. Okay? And so as it turns out, the O has a certain display of sugars, but it's sort of the base. It's sort of every other, somebody brought up, does the O not have anything to it? Yeah, so people that have sort of O blood type, they have kind of the scaffold. They don't really have anything added. So people that have O blood type are displaying a certain antigen that has basically 4 sugars to it, but there's really nothing added to it. People that have A blood, okay, have the sugar, and then what that sugar is bound to, make sure I get this right. Man, I'm screwing up these. Which sugar is which? Okay, so for A, it's going to be N-acetyl galactosamine. Okay, and this is an amino sugar, galactosamine. And so the basic sugar is this one here. So again, don't worry too much about like scribbling this down. I'm just trying to give you guys a feel for this stuff. So that's what that is. So it would be attached right at the anomeric carbon, and so it has the sugar, but then it also displays this extra guy there. Okay. The B antigen, on the other hand, has same thing, sugar. But then what it's bound to is not the galactosamine, but the glucosamine. Okay. So in the glucosamine, okay, and this is basically the exact same sugar as this, except that there's one change in stereochemistry. Okay, so it's a very similar looking molecule. Except this position here is sort of equatorial. Right? And then as you might imagine, right, the AB has both of these, right? So AB has some sugars that have B and some sugars that have A, right? And so as you can imagine, right, the sort of universal blood type in all of this, right, is going to be people. So if you have So if you have red blood cells, right, we'll just say A antigen, B antigen, A and B antigen, neither antigen. So if you think about the antibodies, right, that would be associated with these, right? If you're of A blood type and your cells have the A antigen, then that means that what they're going to do is they're going to recognize the B antigen as foreign. So anti-B, we'll say antibodies. Right? So if you're of the class A blood type, you want to get we'll go through it completely. Right? But if you have the B right, then what you're going to have is anti-A antibodies. Down here, you're going to have neither, and over here, you're going to have both, right? Because if you're displaying both the A and B antigens, then your body's going to recognize those as being native, and so you're not going to have an immune response based against them. Now, if you don't have either antigen, then what that means is that if you see a cell that has the A antigen, or if it has the B antigen on those cells, then suddenly you're going to have a sort of a significant immune response to those. Right? And so obviously this is like really important again. So if you're of the A blood type, right, you'll either need the A, right, or the O type would be fine. That's why the sort of O type is the universal one, right? You can use O blood for anybody because it's not displaying anything that's going to be sort of used against it. Meanwhile, if you're the sort of AB blood type, you can really only give your blood to people that have, you can only give your blood to people that sort of, right, that sort of have AB. And sort of likewise with the others, right? If you have A blood type, you'll only want to give your blood to people that have the A, and if you're B blood type, you'll only want to give to the B. But again, in the O, that's why anybody can give O. So I thought this was kind of interesting. It sort of puts into perspective the sort of immune response. Again, this is something that you guys sort of know about, at least in terms of have heard about this sort of what type of blood I am, and this is sort of why it's important, right? And it all has to do with this sort of sugar sort of epitopes. Okay? All right, so taking this a step further, now I'm going to go into some sort of research that I was involved with during my postdoc that involved sugars as well. And so it turns out that, so this is sort of within humans. But as you might imagine, again, other organisms may have sort of antigens that are sort of native to all of us, okay? And one of these antigens is called alpha-gal, okay? And alpha-gal is kind of important. Okay. So alpha-gal. So alpha-gal is a disaccharide. Okay, so this is alpha-gal. Okay, it's a disaccharide. And it turns out that alpha-gal is an antigen that's basically displayed on the cells of almost every organism out there, with the exception of pretty much us and like old world, like monkeys, basically our direct evolutionary ancestors. So basically everything that's out there sort of has these. What that means is sort of two things. And when I say everything, we're talking about bacteria, for example, the flora, the sort of bacteria that are always in you display this antigen, yet we don't see it as native, even though we've got the bacteria in us constantly. We see it as foreign, but it's sort of regulated in a subtle way within our guts because the bacteria have displayed this, we've generated an immune response against it, and so our bacteria that are sort of helpful to us are sort of regulated to a certain extent based on this. Now what that means is, and we've sort of gone away from this a little bit with surgery, but one of the big issues in surgery, right, is that if you're trying to do sort of trans-species implants, like let's say you try to get a heart transplant from some other organism or some type of other organ transplant from another organism, there's a major problem with that, and that's that organism is going to be displaying this antigen. And so a lot of times sort of organ rejection of the host immune system is generally because of sort of alpha-gal. And so that's sort of interesting. So a lot of times what people have to do is if they have this sort of this antigen in it. They might need to have some sort of enzyme that might break it down. And I think that people have worked on this a little bit so that when you do sort of organ transplants, you don't suddenly go into anaphylactic shock and die, right? Or it would reject the organ and just have it not work. Okay. Now, this sort of idea of having sort of an immune system within you that's based upon sugars and that's sort of ready-made to sort of go in and start attacking something is something that during my postdoc we sort of became interested in. And that's that if you could sort of design and synthesize a small molecule that would, say, for example, bind to a cell selectively that was pathogenic, right? And at the same time, could you sort of display a group that your body would recognize as being foreign, then effectively what you can now do is, say, for example, take a prostate cancer cell for all the men, right? What you can imagine doing is taking a prostate cancer cell, and if there's a protein that's on the surface that's overexpressed specifically towards that cell type, then what you can do is you can build a molecule that might bind to it and effectively label this sort of cell that your body doesn't recognize as foreign and now label it as foreign so that your immune system can now go and start targeting the cancer cell. And so this has been this sort of idea that my postdoctoral advisor sort of had and has really been working on a lot, but certainly there are other groups as well. And so one of the things that people are now trying to do is identify different protein targets or enzymatic targets displayed on the surface of different pathogenic cells, and then trying to engineer molecules that can sort of do this. And in fact, in our lab, we sort of did this with a slightly different antigen, a dimethylphenyl group, because it was more simple to make. But at the time, at our studies, what we weren't able to do, actually I'll just tell you what we did. So you know that about alpha-gal. And so I'll just tie in a couple aspects here. So what I had done during my postdoc work, really the big project that I was sort of successful at, was we developed this molecule, the synthesis of this molecule. To get the sort of stereochemistry to it. But the sort of idea was is that we had again, there might be some things wrong about this structure, very wrong there. And then we had a tether. At the beginning of our work, what we had was this. We had a dinitrophenyl. Okay, so this sort of dinitrophenyl group also was an antigen that the human population actually, we have antibodies already within our system that will recognize this, at least so we thought. They're there, we've seen them as well, but the concentration of them, what we found is that when this would work, when we would have, say for example, we would dope in antibodies, so we'd run these bioassays and we'd dope in an antibody that recognized this to do what we wanted to do. What we found was that when we were trying to do it with human serum, we would get a little bit of mixed results. On occasion, it might look like it was working. Other times, it wouldn't work out that well. After we sort of published the first paper where we sort of showed that this in principle could work, this bound to prostate cancer cells specifically, what we were interested in is whether or not we could attach this sort of alpha-gal epitope to it. Now, what I should point out is that the sort of binding affinity towards the alpha-gal itself isn't generally that high, but generally there needs to be some type of other sugar as well. So this is the alpha-gal, but generally there needs to be some sort of other monosaccharide to get it to build. So basically what this came is we went from having to put on this little dinitrophenyl group to having to put on basically what's going to amount to is a trisaccharide. And the precedents that we had to work with, she's a big deal. Her name's Laura Kiesling. She's at Wisconsin. She works on a lot of sugar chemistry, and she had done a similar thing with a cancer epitope with sort of these integrin binding molecules. So we were working on a different molecule here, but she had something else over here. Oh, sorry, this one bad. Not that you guys are going to look into that, and this is like two. But what she did was she had a different group over here that we had, so she was targeting a different protein that was expressed on different cells. And what she had done was she found that if she put this sort of alpha-gal binding group onto her molecules, that now she was actually able to get really nice data just using human serum. So she wouldn't have to dope in the antibodies like we were doing. So we'd finished the first paper, and, I was a postdoc. In terms of the antibody recruiting project at the time, I certainly had the most synthetic chemistry experience. So I sort of took up the task of going ahead and synthesizing this sugar. And the overall steps, to just give you guys a heads up, the whole synthesis ended up being like 17 steps, right? So you can imagine what a beast of a synthesis this is. But as you'll see, and actually when I went out to give job talks and we discussed this a little bit, I would sort of use this to sort of illustrate how within sugar chemistry we're still really bad at making molecules. So the way that we went about synthesizing this molecule, again, we had a prep. Laura Kiesling had made this in the past. Still, doing a 17-step synthesis when it's the first time you're doing it can sort of be challenging, because you have to sort of work out a lot of the chemistry yourself. Oh, it's right here. Okay, so what we did, and what Laura Kiesling had sort of shown, is that to make the sugar, and the fact that you want to make it as a disaccharide, what you can basically do is you can take this sugar here, okay, and what we're going to effectively do is combine it with lactose. Okay, so this is lactose. The other sugar is, I think, I'm trying to think what the name of that is. This is mannose, I think? Forget. But basically, these are two sugars that you could buy. But what we want to do is we want to make a connection between this sugar and here. This is where we want to make that sort of glycosidic linkage. And again, you can buy tons of this, you can buy tons of this, and all that we wanted to do was make this bond and set this molecule up such that we could make an attachment over here. Okay, so really two productive groups here. We can take this alcohol, this anomeric position, selectively couple it to this alcohol right here, right? And then take this group and sort of functionalize it over on that end. Now we have our molecule that we can start building on. So really just two productive bonds. But this is where sugar chemistry really struggles, is that I just told you guys earlier it's going to be like a 17-step synthesis, because there's a lot of crazy sort of things that you need to do to actually get this to work. And so again, I'm going to show you some chemistry, some of it you'll recognize. There's one specific thing that I think is actually really cool that you won't be responsible for, but I think it'll put into perspective the sort of chemistry behind this. But hopefully the point that you'll see is that sort of synthesis, anytime you ever hear somebody talk about how synthesis is sort of a solved field, all you'll need to do is remember this part of this lecture today to see that clearly molecules as important as sugars even are not a solved field. Okay? So when you look at this overall process, right, this shouldn't be too much of a problem, right? Making this linkage over here isn't going to be too much of an issue, right? Because as we saw previously, if you can just simply take an alcohol like methanol or something like that, and you can treat it under acidic conditions, you'll make that glycosidic linkage. The challenge in this overall synthesis is how do you take a structure with all of these potential alcohols on here and specifically set it up so that you can only react one of them? Right? That's like a huge, I think, endeavor, and it's a huge challenge, and there ends up being a lot of sort of synthesis that you need to do to sort of do this. Okay? So to get this one set up, it turns out that what you do in general is really just a three-step process, and then I'll focus most of my time over here. What we're going to do is we're going to synthesize this molecule here. Okay, so I'll go through the synthesis of this unit first. So the first thing that we're going to do is we're going to take this sugar, and what we're going to do is we're going to protect all the alcohols, right? And we're going to have this sort of thiophenol linkage here. So this is simply going to serve as, you guys saw, like acid right conditions, be good leaving groups. This is certainly going to serve, this is just going to serve as our group for sort of leaving groups. So even this doesn't look like that complicated, but within sugar chemistry it ends up being a little bit difficult. So I'll start with this and then I'll move on to that, just because I can show you how this little modification actually takes some time. Okay? So when you're doing sugar chemistry, oftentimes one of the first tricks that you'll do is if you want to do chemistry at the anomeric position, one of the tricks that people often do is they'll take that molecule and they'll first just treat it to acetic anhydride pyridine. Yes? It's a thiophenol, so sulfur phenyl. Sulfur attached to phenyl.
So why is it
What's a better leaving group? What's stabilizing an anion better? Phenol or ethanol? Phenol. Now you go to sulfur, it's even better because it's bigger, so it's stabilizing even better. So it's just a good leaving group that also coordinates pretty nicely to certain metals that facilitates some elimination. So it's just a leaving group that you'll see every once in a while. That's not a crazy. It's not like a halide in terms of the amount, how fast it'll leave. So it's a little bit more stable, but you can coax it to leave fairly easily. Okay. Now, generally, if you take these sugars and you treat them to acetic anhydride, what you're going to do is you're going to do a global sort of acylation process. Okay. And when you do this, what you're left with is a molecule that has a sort of, at the anomeric position, you have this acyl group here. Okay? The nice thing about this acyl group here is that by simply activating it just a little bit, what you can actually do is facilitate it to sort of leave, right? Because if you've got this sort of acyl group here, what you can now do is treat it with a Lewis acid, right? So if you treat that with a Lewis acid, what that's going to allow is that you can actually leave that group really readily. So having that acyl group, this is one of the first things that's often done in sugar chemistry if you're trying to do glycosidic linkages in an actual synthesis, is you'll acylate the whole molecule. And then what you'll end up doing is you'll sort of treat it to something like a Lewis acid. I think in the case when we did it, it was BF3 ether 8. And in this case, I don't remember if there was an intermediate, but so it might have been slightly different than this, but we'll just say trap with thiophenol. I'm not sure if this is exactly what we did, but at least it will serve the purpose. Okay? Okay. Now the problem is, right, that oftentimes these acyl groups, as we'll see, so you can imagine taking this and then going ahead and starting the chemistry, but because of some downstream issue, we then had to take this molecule, deacylate it, so basically pluck those off. Conditions were probably, what would you think? How would you hydrolyze acyl groups? H plus and what? Water, maybe? Yeah, I think it might have just been, acetic acid water. It might have also been like basic as well. I'm not sure exactly what the conditions were. But yeah, you're going to do hydrolysis to sort of generate the free alcohols. And then you're going to benzylate. So you're going to deprotonate all those alcohols with sodium hydride and then flop it back on. So again, the overall process to get to this compound here, as you sort of see, right, is really the long process. Really all we're trying to do is protect it and put a group on there. But again, because of some of these subtleties within sugar chemistry, we had to acylate it, so that then we could do our modification, so then we could deacylate it, so that then we could effectively put on the protecting group at the end of the day that we want. And this is super common issue with sugar chemistry, and it's probably one of the things that adds up so many steps in sugar chemistry, is that there's a lot of, okay, I've got this protecting group because I need this for this step, and I need to swap it out for this protecting group for this step. And so this is a big issue with sugar chemistry. So you can clearly see that really we haven't done much, but it took us 4 steps, right? So you think about that, that's 4 reactions, that's 4 purifications, that's 4 isolations, that's 4 NMRs. It's time, right? Simply to do that thing, okay? Now that's easy, right? Doing what we need to do for this sugar is actually a completely different problem and bigger monster of a problem altogether. But it involves a really cool thing within sugar chemistry that I think is kind of sort of noteworthy. Okay. Shoot. Okay. Okay, so I actually didn't write this down, so I'm going a little bit on memory here as well, but I'm pretty sure I remember what I did. So, here's the sugar that we have. Okay. Now, the issue with this compound in general, for first and foremost, is that when you're going to start doing chemistry on this, what you need is this group, because I definitely don't have it written down. Okay, so what's going to happen is, as we discussed earlier, we basically have what amounts to a protected aldehyde, right? And that protected aldehyde is going to cause a lot of problems if we're trying to do any type of chemistry. We treat the aldehyde with a nucleophile, it's going to react. So the first thing that we need to do in the synthesis is protect that as effectively a group that we're going to want. And what we want to protect it as is, in fact, this sort of benzyl alcohol, right? Because we had seen previously that we're going to put benzyl groups on this. So what we're going to do is we're going to put a benzyl group on there, because at some point we're going to actually have it globally benzyl protected. And so to get this molecule to just have an OBN here, right? What we're going to do is that same sequence that we did before, but instead of using thiophenol, right, we're going to use benzyl alcohol. So what we'll do is we'll start by doing acetic anhydride, pyridine, right? Then we're going to treat it to a Lewis acid and benzyl alcohol, or sorry, phenol, or benzyl alcohol, excuse me. So now benzyl alcohol is going to add in there. And then what we're going to do is hydrolyze at the end. So our first step is effectively a three-step process, simply to protect one group out of there. Okay? So that's the first step. This is going to take me more time to draw this. And everything else is hydroxyl. Now this is what's cool in this synthesis, which is what I thought was pretty awesome. Okay? In this molecule, you have a whole bunch of different alcohols. And this next reaction that we did, I thought you guys will be able to appreciate to a certain extent, based upon what you've learned in organic chemistry, and sort of understand how neat it is, right? So, this is equatorial. Didn't draw it that well. You've got all these alcohols in here, and what you want to be able to do is you want to be able to selectively react one of them. And what they did, and what I did, following what somebody else had in the literature, which I thought was pretty neat, is that when you look at the orientation of all the alcohols in these molecules, you'll notice that a lot of them, as you're going around here, are sort of equatorial, right? But what that means is equatorial, it means one up, one down, right? Right over here you have this alcohol, but over here you have one down, one up, and you have this other one that's one up, okay? So you got these two groups, in this case, that sort of goes equatorial, axial. But what that means is that these two groups, if you think about them in a sort of a cyclic system, is they're both syn to one another, right? So just to sort of simplify it, again, I'll leave out the rest, but this is what I mean, is that if you sort of drew in a flat conformation, they're both going to be in the sort of same direction. What that means is that you can actually take a molecule like this, and you can have it wrap into a 5-membered ring. You can't do that with these other groups, so let's just... I'll just put an X there, okay? But you can sort of see that that's a 6-5 ring system, right? If you had a trans 6-5 ring system, that's not going to be able to form because it's way too highly strained. Does everybody see that? If this was trans, that would be highly unstable and highly strained. But because of the fact that they're syn to one another, you can actually form that sort of 5-member ring there. It's the only position, the only two alcohols on this entire molecule that have that within them, okay? So what that means is we can sort of leverage that. And the way that people have worked this out in sugar chemistry is pretty neat. So if you go ahead and you take this molecule and you treat it with, I think the reagent was a tin reagent, it was a tin oxide. I think it was dimethyl tin oxide and some acid. I could be wrong on what the alpha groups are. It was a tin oxide, okay? And it was acting effectively like sort of an acetal formation, right? So if you think about acetone condensing, so that same type of thing is like acetone condensing, except that instead of it being the carbon, it was instead tin, okay? And what that allowed was us to basically generate this stannate. Okay, so it effectively allowed us to make that standing right there. Okay? Now, what's interesting about this is the moment that you chelate those oxygens to a metal, right, those oxygens effectively become, that tin oxygen bond effectively becomes much more nucleophilic. So it's kind of analogous to making, it's sort of the oxygen equivalent to like almost like Grignard in many ways. It's not specifically, it's not that stable, but the point is that these bonds are now more nucleophilic. Okay? So we basically taken, and by tying these two oxygens, which are syn to one another, the only two ones that are syn, we've tied that back into this stanocycle. Now we've effectively increased the nucleophilicity of those two oxygens over all the other oxygens in the system. Okay? But that still means that there's two oxygens, right? And so what happens is that when you take this molecule and you treat it to an electrophile, the more sterically accessible alcohol, or the more sterically accessible tin oxide is the one that's going to react. Which one of these two do you think is more sterically accessible? Yeah, the one on the bottom, right? Axial is sort of sticking out, right? Axial is more stable for things to sit in axial. So this becomes the most nucleophilic oxygen in there. So what you do is you then take this molecule and you treat it with allyl bromide. Why allyl bromide will become clear in a minute. But when you take this molecule and you treat it with allyl bromide, it's a good electrophile. But you treat it to this whole system falls apart, and what you can do is effectively get an allyl group at that one position. And you effectively isolate that group. Now we've got an allyl group there, we've got an OH there, and we've got an OH there. Okay? Now an allyl group is not a protecting group that you guys are very familiar with. But it turns out that one of the things that you can do with an allyl group is you can actually isomerize, using mild sort of metal catalyzed process, you can effectively isomerize that bond and make it the enol ether, and then that will hydrolyze off just under acetic acid and water. So you can sort of hydrolyze that off. So what we did was, at this step, we're still not there yet. So all we've done is sort of put a group there. That's one of the things I was like really curious about, right? I'm going to go through all these sort of protections. This is oftentimes how synthetic chemists' mind works, is they say, okay, well, why are you treating it with the allyl group? Why don't you just treat that with the glycosidic molecule that you just had? And I think if somebody could develop that, that would be unbelievable. The thing is that doesn't exist currently, but that could be something that would be a nice sort of breakthrough. But what's going to happen here is we're going to benzylate, we're going to put benzyl protecting groups on all the rest. Right? And then what we're going to do is we're going to deprotect the allyl group. So we'll have benzyl groups on everything, then we'll have the allyl group, and then we're going to deprotect allyl group. Okay? Now what we'll have is our molecule ODN. So now we've globally benzylated this molecule and we have our alcohol free. So at this point, again, we'll just use the glycosidic linkage to effectively make our compound. It's not done at that point. There's some other interesting chemistry that has to go on. But I think the point hopefully has been made, is that within sugar chemistry, there are things that you can do to perhaps gain selectivity versus certain alcohols, but we clearly see that the methods that are out there currently are very time-consuming. Again, all I've done and all I've shown you guys, we've probably gone through like 10 steps there, and all I've shown you is how to prep the two components that we're going to put together. The rest of the chemistry is deprotecting and then putting on the rest of the molecule together. So that's simply to make one carbon-oxygen bond, and this is sort of state-of-the-art chemistry with sugars. So one of the things that people try to do, so 2 points and I'll let you guys go. One real important area, I would say, in terms of sugar chemistry, any of you that are interested specifically in research within, say, for example, biological chemistry, or is to perhaps get enzymes that could do that in a single step. Can you engineer an enzyme that would put those two together for you without having to worry about all those different protecting groups? So sugar chemistry, I think, as a synthetic organic chemistry standpoint, I think represents an area that I'm not sure a synthetic chemist is necessarily going to come up with the answer to, but I think the enzymes, again, because that's how they're actually made in your body, right? If you could harness that same power, these would be valuable here. I'll also point out that I spent time making this molecule, stuck it on my compound, went ahead and tried it with the human serum. So I spent, I think, 3 months making this molecule. Tried it in the human serum, and it didn't work. so there's to go research, right? It's a lot of time, bang your heads against the wall. But what's interesting is that it turned out that there is a single sugar called rhamnose, which actually we have antibodies that are even better for. So it's a single sugar. It makes it much more synthetically readily available. I was actually working on that to a certain extent by the time that I left, but now people are starting to recognize that you don't need alpha-gal. There's other classes of sugars that can get the same type of benefits. Okay, so with that, for those of you who weren't here at the beginning, again, check through your exams. We noticed a couple adding mistakes, so just take a look, police your own exam, just double check if there looks like something that's an adding mistake. Let us know, and with that, I'll see you guys on Wednesday.
Right.